Hello, I'm Les Bennett, representing the American Association of Pharmaceutical Scientists, and I have the pleasure over the years of interviewing outstanding pharmaceutical scientists in a series that we can have a record for future young scientists and older members to recognize those in our profession who have created such an impact. Today, I am very honored to have Professor Nicholas Bodor of the University of Florida as our interviewee. Nick won the AAPS Distinguished Pharmaceutical Scientists Award in the year 2007 and has had many other wonderful experiences and changing aspects related to the pharmaceutical sciences, and today we're going to have the opportunity to explore that with Nick. So Nick, welcome. Let's go back to your history. You went to university in Transylvania and then got your PhD in Cluj. And give us a little history of then what happened, how you got over to the US.
Well, I was working after I got my doctorate in Romania, although I am Hungarian, but that's Transylvania. I worked at the Pharmaceutical Research Institute in Kolozsvár, but you call it Cluj.
Just because I don't know how to pronounce it.
No, it is because you called it Romania. And I worked there in the synthesis of a large number of important new drugs which were introduced then in Romania. The object was to find new ways to manufacture and patent drugs discovered in the West, because at that time the patent laws were different. So that I was doing a set of steroids, antibiotics, and all kinds of drugs. But I was very interested also in theoretical chemistry on the side, and conformation analysis of Derek Barton and Michael Dewar, who was an outstanding, all-time best physical organic chemist, and he published the molecular orbital theory of organic chemistry in the Journal of American Chemical Society. I read it and followed up with some other papers where he wrote about remote effects. And I thought the idea is very nice, but he really didn't demonstrate it well. So I designed some experiments and I published 2 papers which I sent to Michael Dewar, which followed up by an invitation. But to get out from Romania it was impossible. But I don't want to go in those details. But through various circumstances in the end of '68, I managed to come and work with Michael Dewar. I had to go back after 1 year, and he sent another invitation, and I came back, and after that I didn't return to Romania. I couldn't return even now. And
okay, so then what? You then, I know you got to Kansas and Interact.
It was very, collaboration with Michael was very successful. We published in 2 years like 14 major papers in JACS, etc. And then I got married, and I realized that postdocing doesn't give me enough to live on. And I answered an ad from Professor Higuchi's Tech's new company. That time it was ALSA. And he asked me to Lawrence, and he offered immediately a job for me. And I started out at ALSA working on prodrugs. Then he transformed ALSA's Institute of Lawrence into his company called Interex. And I worked there until '78. from 72 to 78.
That's where I had the pleasure of meeting you for the first time.
Yes, well, we shared a room in
That's
the
right.
Higuchi seminars a couple of times. We played tennis, etc. And that was a very rewarding, very successful time of my year. Tech was really wonderful. We had lunch, working lunch for 6 years every day. And then in 76, he decided to bring me with him when to visit Japan. So I can give the lectures and he can do the introductions. And that was really good because that helped me later on develop very important connections with the Japanese scientists. And as you know, Higuchi seminar we went every year, and in early 78 at the Higuchi seminar, Mike Schwartz, who was the dean designee at University of Florida, came to me after I gave a lecture, and he told me that he has a couple of positions for quality improvement at the University of Florida, and would I be interested in. So to make it short, I came for interview. I remember I gave a talk when I introduced myself and apologized for my Texas accent. And soon after he offered the job and the full professorship actually. And then I became chairman of medicinal chemistry and started to work. Immediately, I started in January 1. In February, I submitted an NIH proposal, and as it happened, you were the chairman of the pharmacology group, which awarded the grant for me. That was about the brain targeting, which was then very successful. It became a very important new way, or I say still one of the best ways to target, not just deliver, but target drugs to the brain. To continue on the path of my professional development, I built up a large group at the University of Florida, and after I published in Science some of the brain targeting concepts, a bunch of venture capitalists approached the university. And there was one successful who offered $6 million to the university to license this whole brain targeting and formed a company called Pharmatech out in Gainesville. And so I shared my time between the university laboratories and the Pharmatech laboratories and offices. At some point, I had a group of about 75 people, and many really outstanding people was working with me. And this was the brain targeting, but in the same time, I worked on a parallel drug design project, which I called soft drugs. The idea was to...
We'll come back to that.
Okay.
I want to spend some time on that, but let's just go history-wise.
History-wise.
Yeah.
Okay, in 98, the university had a visitor, famous pharmaceutical entrepreneur, Phil Frost, whom I knew for a long time. I did consulting for them, and it happened I told him that there is a large, very well-developed research institute in Budapest, Hungary, which is up for sale, because with the change of the system, a central research institute for the whole industry lost its basis in the various companies became competitors, so they couldn't have one central research institute. So he said, okay, let's go see it. He had his private plane, that time just a DC-9, and we flew to Budapest. We visited the institute. That time it had about 500 employees, scientists. And he said, I buy it if you manage it. So that put me in a real quandary, because I had there a bunch of my former students, friends, etc., and the institute was 1 month away from bankruptcy. So I accepted the job and became the managing director of that research institute in November 99. Followed that, I accepted the senior vice president and then chief scientific officer of AAPS.
So just to interrupt a little bit. So you had the academic position. Did you take a leave from the academic?
I took a leave of absence, and that was continued, renewed. The university was very supportive, and they gave me leave of absence. And I always thought I went there for a year or two, but then I stayed in Miami even now. I continued to have a group at the University of Florida, but the work we have done at the Research Institute in Budapest was truly exciting and on a grandiose scale. That institute was a fully comprehensive development from the drug discovery to clinical trial. We have done everything. And it was well known in Europe, and I had to go there every month, at least 1 week. And the rest spent with IVEX and some in Gainesville. So that was until IVEX was sold to Taiwan. And then I left. The university called me back. I went back part-time and set up in 2006 my own company in Miami, which is called Bodor Laboratories. And that's where we are working with my son and daughter. We have a number of licenses out, and I have 2 new phase 2 drugs. One which was before, but maybe we talk about that with the sub-drug.
Okay, so let's go back just to the, you are a wonderful example of moving from academia to industry to academia to industry to academia. And why don't you just give our audience, the view of, what are the differences and how do you put it together, and is it difficult to switch back and forth? And
yeah, it's a very interesting question and issue, because drug discovery and development is something which is really not done in academia. Discovery part, yes, and then clinical trials, yes. But what happens in between, academia is not familiar with. That's what the companies do. And I realized that if I really want to have a drug designed by me reaching the market, I have to control basically all of these aspects. So I had in my group people involved in the synthesis, in biological testing, pharmacokinetics, testing, activity, toxicology, etc., because that's the only way you can really push something forward, because the collaboration, particularly in academia, is never, in my mind, never works out right, because we are all prima donnas and want to do things how we like, and maybe you don't like it how I do. So that was the basic thing, and what was helpful, this first investor in 82 who put in $6 million into Pharmatech, that allowed the development company to...
Was Pharmatech also owned partly by the university?
Yes.
Yeah.
20% of the project was owned by the university, and it was really a good arrangement. That happened only because not soon before, as you know, the NIH allowed universities to own patents discovered there. And so that was Pharmatech. Then I got involved through the NIH grants with ophthalmic drugs, targeted ophthalmic drugs and some drugs. And so another company was set up with another group of investors, which called Xenon Vision. Eventually, these two companies merged. Then Pharmos, which was an Israeli company, bought the merged company and continued development of a number of my drugs which were in development, both at Xenon Vision and Pharmatech. And eventually it was successful to the point that Pharmos licensed one of this unique soft steroid to Bosch and Lomb, which put it on the market in '98. And it is a very successful drug. All the years, I think it must have sold about for $2 billion. And a number of products containing glutaprednol are on the market and in development and for other fields as well.
Okay, so I think it's always an interesting question for us academics that also do industry relationship of how we deal with that conflict of interest, because you're working for Pharmatech, you're working for IVACS, and yet you're maintaining a research group in the University of Florida, and how do you handle that, and what do you pay attention to?
Well, you just do both. You know, what happened, I had a large number of students, and one issue is I never allowed students to get involved with companies' research, because the objective of the student is to get a degree to learn, to develop something, even if scientifically interesting studies, but if it's a new drug in development, it has to be done by technical people related to the company. And that's what happened. Actually, they were physically separated. The Pharmatech was in Alachua, which is about 25 miles from Gainesville. And I had very good people. You know, when Pharmatech was opened, I was successful to attract one of my first students, Ephraim Sheck, who became vice president there, then patent agent, and many others. One of the investors became president of the company, and the university allowed me to act there as vice president of research, and I spent maybe 20% of my time there. The university, particularly some people, were very eager to hold the loop on these university interactions. So everything was up and up.
Okay, good. So let's go back and talk about retrometabolic and soft drugs and your development there, which has certainly been changing of the environment in the field. Give us a little background on that.
How the soft drugs came up at Interex, I was working with chloramines, which big named soft chloramines because they were not irritating and based on amino acids, and we call these soft agents. And the fact that they cleave by design to inactive or non-toxic material, I later borrowed the term and used, and first I published on the subdrug concept in 1980. I published 3 papers in a row in JMed Chem about the concept, the classes, what you can have, and Among these, the most successful are the inactive metabolite approach and the soft analog. And various applications were developed by us and then other people, and very satisfying to me in the past year or so. Many companies are working, and they acknowledge in the publication the soft drugs in a variety of fields. Universities and companies, and some major company has even a dedicated group for soft drug design. And so the main thing is to design the toxicity, or the lack of toxicity actually, into the structure. So a soft drug is a, let's say, isosteric, isoelectronic analog of a known active drug, but there is a weak spot built in the molecule, which by design, most of the time, sensitive to hydrolytic metabolism, not oxidative. In this way, we direct the usual, mostly oxidative metabolism of a drug into hydrolytic, which is non-saturable. The esterases are abundant. And this was really very successful in a variety of fields. And I hope that a number of subdrugs will be developed by other people, because we are working on some in the new company as well. That was the subdrug aspect. The other side of the retrometabolic design is where an active drug is transformed chemically into an inactive construct in such a way that predicted metabolism sequentially modifies the physical chemical properties, which then influences the distribution and ultimately yields delivery of the active drug only to the target of the target organ. For the brain targeting, the simplest was, let's say, estradiol to target to the brain. You hook it with dihydrotrigonelline, dihydropyridine structure, which is lipophilic, penetrates the brain. Then the NAD/NADH system, which is ubiquitous, converts it to a positively charged quaternary salt, which gets locked in the brain. In other words, we are using the blood-brain barrier from inside. Now, what is oxidized outside, it cannot go in. What's inside cannot come out. Stays there, the other is eliminated. Then what is in the brain, it is cleaved and slowly releases the active drug. I mentioned estradiol because that was very much studied, including phase 2 human trials. And after one single dose, you can see estrogenic activity without high blood levels for 1 month. It's a very interesting method how to separate basically the brain from the body. And this we applied a variety of other compounds, from anti-tumor agents and antibiotics and antiviral agents, etc.
So, thank you for that description. Now, you're the envy of many pharmaceutical scientists, because not your grandchildren, but your children actually have gone into the field. So you have a son who's a professor, and your son and daughter are also working in the company.
Yes,
and
yes.
so what's the secret? How did you get them to be interested in pharmaceutical sciences?
Well, first, I got them interested in education. I am happy that all 3 of my children have doctorates, and they are committed in their field. My oldest son, he's mostly practicing physician, professor in the medical school. But my younger son got his PhD in pharmacology. Then went to Heidelberg. He was a Humboldt Fellow for 2 years. Then he went to Columbia and got an MBA. So he's well prepared for the industry. And I am lucky, really lucky, that he always thought that what I am doing is very interesting and important, and I could convince him to stay with me. Maybe I am a little selfish in that because he's very good, but he's very important for the company. My daughter, who got a doctorate in psychology, she's also involved with the company, mostly with the intellectual property aspect.
Okay. So what do you see, well, let's talk a little bit about your important member of ACS because of medicinal chemists, and an important member and well-recognized member of AAPS. What do you feel this, the synergy that goes along in the two disciplines, and any things that really sort of push them apart? Or
that is an interesting question you are asking, because you are well aware situation. Really, the American Chemical Society and the medicinal chemistry section itself is very large and very different. The medicinal chemistry in AAPS is, let me say, very weak. It is involved mostly marginal things, and really the main discovery and design function for drugs takes place mostly in the industry by excellent medicinal chemists. And it was, and it still is somewhat difficult, to go to both organizations and participate. I was, as you know, really proud to be inducted in the Hall of Fame of the American Chemical Society.
This
and
just
on the other
happened
hand,
2 years ago.
2 years ago.
Yeah.
And on the other hand, in AAPS, in AAPS I was more active, although I did participate in a number of symposia in the American Chemical Society. The main problem, as I see it, that AAPS and the pharmaceutical scientists who are members here are doing mostly what I call peripheral research, nothing basic. It's the methods they use, with the few important exceptions who establish basic rules, but most of the others in the pharmaceutical schools are doing studies for somebody. Related to medical school, not too much with the chemistry, really. And maybe something should be done about that. Actually, it would be really good, and somebody like you, who is the most important person in this organization, could do something that, like the academy or the Institute of Medicine, to pay more attention or to form a section of pharmaceutical sciences.
Hard to do.
Yes.
Yeah, we're a discipline that is a growing discipline. I mean, we've formed AAPS to give the recognition to the pharmaceutical scientists. And it's hard to break those barriers and stuff, but I think we're succeeding, but it takes time.
Yeah,
it takes time.
Other similar organizations like the Hungarian Academy, they do differently.
Yeah.
But
good. So I remember you were a vicious tennis player when we used to play. Are you still active playing tennis?
Not anymore.
You have knee problems.
I have knee problems. However, in my company's office, we have one large room dedicated to a ping pong table. So at lunchtime, Eric and I, we play ping pong many times.
How old are you, Nick?
I will be 76 in
Okay, so
February.
he's a young guy. He's a year younger than me.
Yes.
So what's your plans? What are you going to try to do in the next few years?
I really try to get at least one of these new drugs I have phase 2 and the proof of concept went through. I am very excited about it. You know, people are greedy. I had one drug which I designed which got on the market, and some other I was contributed to, but I have now at least one other I would like to get it to the market. And I am more pragmatic now, you know, I don't do research on the side, except I have collaborations with a number of academic institutions, but we work mostly with CROs in the development.
Well, Nick, thank you for sharing your history and your vision, and we're proud of you being a member of AAPS and being our distinguished scientist, and I know the chemists are proud of you in terms of the Hall of Fame and your honorary degrees and everything else, and thank you so much for sharing your opinions with us today.
Thank you very much. Thank you. Enjoyed it.